Hello and welcome to AP Bio Simplified. Today we're doing the math review. Since math is all over AP Biology, we needed to analyze experiments and data, and for the exam we'll need it for the free response questions and the multiple choices one. And today I'm going to show these 6 common topics. First one is chi-square. Chi-square test helps you decide if your results match your expectations. The formula is chi-squared equals the sum of observed minus expected squared divided by expected. And using this, we can get the chi-square value. In order to get the critical value, we will look at this table. The degrees of freedom is the number of possible outcomes minus 1, and we normally use a p-value of 0.05. So for example, if I have 3 possible outcomes, my degrees of freedom will be 2. And using a table, we'll get a critical value of 5.99. Now we can compare this value with the chi-square value. If the chi-square value is greater than the critical value, we can reject the null hypothesis because the difference is statistically significant. And on the other hand, if chi-square value is less than the critical value, we fail to reject the null hypothesis, and the difference could be due to chance. Alright, next up is Hardy-Weinberg. The equations are p plus q equals 1 for alleles, and p squared plus 2pq plus q squared equals 1 for genotypes. And there are some assumptions, including there should be no mutation, no gene flow, no natural selection, there should be random mating, and a really large population size. Now let's do a quick example. If 9% of the population is homozygous recessive, what percent of the population is heterozygous? So since q squared we know is 9%, q will equals to 0.3. And since p plus q equals 1, p is 0.7, and heterozygous is 2pq, so we will get 42% are heterozygous. And water potential, this one looks a little bit more complex, but it's just actually plugging in numbers. So water potential equals to pressure potential plus solute potential. But in open containers, pressure potential equals to 0, so the total water potential basically equals to solute potential. And to calculate the solute potential, we use this equation, negative ICRT. I is the ionization constant. So for things that do not ionize, like sucrose, it's 1. And C is the molar concentration, R is the constant, and T is the temperature in Kelvin. After we get the water potential of different solutions, we can know where water moves. So water will move from the high water potential to the low water potential. And this also means that water will move from less concentrated solutions to more concentrated solutions. So surface area to volume ratio, this one tells us why do cells stay so small. Because small cells will have more surface area for each unit of volume, and this will be better for diffusion and transport. And here are some equations for calculating surface area and volume. They are all on the equation sheet in AP Bio. And now a quick example. Let's compare 2 cubes. The 1 cm cube has a surface area of 6, volume of 1, so the ratio will be 6. The 3 cm cube has surface area of 54, volume of 27, so the ratio is 2. This shows that the smaller cube has 3 times higher surface area per volume. This could mean that the smaller cube has 3 times better diffusion efficiency. Now, time to model populations. Here are 3 equations. The basic one is just birth rate minus death rate. The exponential one is Rmax, which is the max per capita growth rate times the population size. And logistic growth is a little bit more complicated, so let's do an example. And if now this population has 100 individuals, Rmax equals 2.2, and the K is 500, now we can plug into these numbers into the equation. So we'll get 16 individuals per unit time. Another thing that we need to know for AP Bio is the graph of exponential and logistic growth. The exponential graph will be J-shaped and the logistic will be S-shaped because it will reach a carrying capacity. Lastly, the Simpson's diversity index. This formula measures both richness and evenness. So we will need to take into account of the number of individuals of each species and the total number of all species. And after we get the results, if it's closer to 1, it will be more diverse. Now, in a sample with 4 species and counts of 10, 52, and 13, after plugging into the equation, we get that index is 0.507. This shows that there will be lower diversity due to the dominance by one species. Yeah, let's see what you remember. Pause here and try these 5 practice questions. You can use your formulas and don't forget your calculator. Here are the answer keys. Alright, thanks for reviewing with me. If this helped, like and subscribe, and leave a comment if there's another topic you want me to explain next. See you next time.